Hello, everybody, and welcome back to BioSC 140 discussion video for Lab 13 and 14, EMG Biofeedback. So you should have already watched the experiment video for this lab. Let's get into it. So a little bit of review. Know your AKAs. EMG is an electromyogram. Electro meaning electricity, myo meaning muscle, gram meaning like graph or recording, electrical muscle recording. What does it measure? It measures electrical activity or action potentials, not strength. So electrical activity and strength are related, but they're not the same thing. Generally, the more action potentials or the more electrical activity you have, the more strength you have. Now, you've seen the setup, you've seen the pads that we put on our subject's forearms for this experiment. Those pads, the EMG, records all electrical activity. This could be electrical activity due to nerves. It could also be electrical activity due to muscles. Both will show up on the EMG. All right, so question. What is happening when you squeeze harder? What is happening in your muscles when you squeeze harder? So let's think about the procedure that we did for this experiment. We recorded the electrical activity in a person's forearm, and we had them squeeze their fist with 25% effort, 50% effort, 75% effort, 100% effort. What changed? What was happening when the person squeezed harder and harder? Well, let's talk a little bit more about some background information before we really jump into our results and what we saw in this experiment. So, when a nerve sends a signal, so I'm going to repeat this throughout the semester, but get it now because it's a little against what I think most people think. It's a little tough to wrap your mind around, but we're going to get it. We got this. When a nerve sends a signal, it's the same strength of signal every single time. So nerves are famous for sending signals, electrical signals, down this long arm called an axon. Every time that signal is sent, that signal is called an action potential. Every time that action potential happens in a nerve cell, the strength of the signal is the exact same. So right here, you can see this electrical recording of action potentials. Each one of these peaks is an action potential happening. What do you notice about the amplitude, about how strong each action potential was? Every action potential has the same strength. Now we're going to get into why this is when we talk about nerves in about 3 weeks. But you need to just accept it for now. We'll get into why later. Every action potential that a nerve sends is going to have the same amplitude, the same strength. Look down here. Down here, we have the same thing, electrical recording of a nerve sending action potentials, sending signals. The intensity of each signal, the amplitude of each signal is the exact same every single time. So, how is information coded? How do nerves change the signals that they're sent? You know, how do nerves vary the signal that they send if they can't change the intensity of that electrical signal? Well, there's two ways that nerve signals, that nerve cells send different messages, that they modify the messages that they send, that they encode the messages that they sent. They cannot use amplitude to encode different messages. So they use something called spatial summation and temporal summation. Spatial summation means that you have increased recruitment or an increased number of active units. An increased number of active units. So if you look at our photo up here, spatial summation is when motor unit 1 and motor unit 2, these nerve signals, send signals at the exact same time. So if you want to send a large signal and you're a nerve cell, you cannot increase the amplitude of one single signal. So what can you do? You can use spatial summation. You can have multiple nerves send signals to the same area at the same time. So increased recruitment, increase the number of active units for nerves. Temporal summation means you increase the frequency of one individual fiber. So once again, you cannot increase the amplitude, the strength of a single signal. So if you want to send a big message, you just increase the frequency at which you send that message. If you want to decrease the intensity of your message, you can decrease the frequency. That's temporal summation. Now, muscles are arranged in bundles. We have all these muscle fibers within one muscle. Within our bicep, we have many of these muscle fibers. If you want to lift up something that weighs 1 pound, you don't need to use every single one of your muscle fibers to lift up that 1 pound weight. It doesn't. Your body only recruits a smaller number. Now let's say you're trying to lift up a 50-pound weight. Well, that's going to require more muscle fibers. It's going to require more recruitment. You have to spatially summate those muscle fibers. You have to get them to work together at the same time. You have to use spatial summation of muscle fibers in order to lift that 50-pound weight. These are examples of spatial summation. Now, let's use some analogies. So, if you want to light up a room, you could have one really bright light, or you can use spatial summation. You can use many lights sending the signal, recruit many lights, and you can have a lit up room. Or you can do temporal summation. Temporal summation is more like using one of these sliding knobs or turning knobs, where the rate at which photons leave the light can increase. You can increase the frequency at which photons leave the light. So temporal means like time related, more photons leaving within a given time. So increased number of action potentials for each nerve is going to be temporal. Spatial is increase the number of nerves, increase the number of muscle fibers activated at one given time. All right, so dominant versus non-dominant. Let's hypothesize. Which arm do you think had a higher amount of electrical activity on the EMG readings? What about for 25%, 50%, 75%, 100%? All right. Which arm do you think was stronger, the dominant or the non-dominant? Well, right here's some typical data. Usually, attempt number 1, 2, and 3 are very close together, are pretty close together for amount of electrical activity. When doing 25%, 50%, 75%, they're usually pretty close to the amount of electrical activity happening for each attempt. When max effort is used, there's typically more electrical activity in the non-dominant arm. So typically the non-dominant arm has more electrical activity during max amount of effort. Why is that? We'll get to that. Which arm do you think is stronger, a dominant arm or non-dominant arm? Typically, people are able to generate more force with their dominant arm. I know for me, my right arm is much stronger than my left arm. I'm a right-hander. I'm right arm dominant. Makes sense. So, why is that? This is because we use our dominant hand more often, and the nerves and the muscle fibers become more efficient. They don't need as many signals to generate maximum amount of force. Less familiar pathways require more signals for contraction. So your dominant arm generates more force with less electrical activity because those electrical pathways, those nerve pathways are more efficient. Those muscles, you don't need to recruit as many muscle fibers. It's more efficient. Your non-dominant arm generally has more electrical activity, even though it generates less force because it's less efficient with that electrical activity. All right, calculate percent increases. Perfect. All right, let's move on to biofeedback. So let's talk a little bit about our hearts. So we mentioned, actually, I'm not sure if we mentioned this. I don't think we mentioned this yet. All right, so heart rate. Within our hearts, we have something called the SA node. The SA node has cells in it that are designed to initiate a heartbeat. So the SA node has cells that initiate each heartbeat. And they can initiate each heartbeat with no input from the outside world. If the SA node stops receiving signals from the brain, so no nerve signals go to the SA node, and if then it receive no blood that's carrying hormones. So no hormones, no nerve signals, no outside signals go to the SA node. The SA node will still generate heartbeats at a regular interval. They're considered autorhythmic cells in the SA node. Now, these autorhythmic cells do receive messages from your nervous system, and they do receive messages via hormones in your blood. So that rate at which the autorhythmic cells generate the heartbeat, they generate a heartbeat by themselves. They don't need outside signals, but outside signals affect how fast that heartbeat happens. Electrodermal activity, or EDA, we also call it galvanic skin response, GSR, is controlled by the autonomic nervous system. And I said that it measures the ability of electricity to travel over your skin. Well, I also said to hypothesize what might affect it. Well, sweat. Electricity moves through salty water really well. So perspiration or sweat affects your GSR, it affects your EDA. So it's a good way to measure the amount of sweat that a person is producing. So you measure GSR, you measure GDA, you can measure the amount of sweat a person is producing. Sweat glands are controlled by the autonomic nervous system. So let's look a little bit about the general format of the nervous system. We're going to get some depth in lecture in a few weeks. Let's just kind of check it out. We have the nervous system as a whole, and the nervous system breaks up into a pair of things: your central nervous system and your peripheral nervous system. Your central nervous system is your brain and spinal cord. Your peripheral nervous system is everything outside your brain and spinal cord. And your peripheral nervous system falls into two categories: afferent or efferent. Afferent nerve fibers send messages towards your central nervous system. Efferent nerves send messages away from your central nervous system. Efferent nerves effect change. So afferent nerves are sensory nerves. They send messages to your central nervous system. Efferent nerves affect change. They send messages to things that away from your central nervous system to things that can affect change, like sweat glands, like your heart, like muscles. Within your efferent branch, you have two categories. You have your somatic, which are going to be your voluntary muscles, your biceps, your triceps, your quadriceps, and you have your autonomic nervous system. Your autonomic nervous system works below the level of consciousness. Can you think really hard and make your heart beat? No. Can you think really hard and constrict a certain blood vessel? No, it's below the level of consciousness. It happens automatically. That's your autonomic nervous system. Your autonomic nervous system is divided into two main branches, your sympathetic and your parasympathetic. Have you ever heard of fight or flight? Fight or flight? Well, when you get ready for like a sporting event, when you get scared, when you get ready to fight or run from something scary, that's when your sympathetic nervous system kind of takes over. Those types of activities are controlled by your sympathetic nervous system. When you want to rest and digest, those activities, resting and digesting, are mainly controlled by your parasympathetic nervous system. We're really going to get into these a lot when we talk about the nervous system. All right, so central nervous system, aka CNS. Know the AKAs. It's made up of your brain and spinal cord. Your brain and spinal cord are the information processing centers of your body. Your brain and spinal cord process information and decide what to do with that information. Your peripheral nervous system, your PNS, is everything outside your central nervous system. Afferent nerves are sensory nerves, and they send information to your central nervous system for processing. Efferent nerves are how your central nervous system affects change. When your central nervous system decides it wants to do something, it wants to change something, your efferent nervous system is how it makes those changes. Information delivery is your peripheral nervous system. From senses, from your senses to your central nervous system, or from your central nervous system to things that can affect change, like muscles or sweat glands. Let's look at this flow of information real quick. This is the general flow of information in our sensory and our nervous system. We have a stimuli. That stimuli is picked up by a receptor. That receptor then relays then this afferent nerve relays that receptor information to your central nervous system, your brain and spinal cord. Your brain and spinal cord decide what to do, and it decides to affect change. It affects change using efferent motor neurons. It could use the somatic system, the skeletal muscles, or it could use the autonomic nervous system. Now, within the autonomic nervous system, it could use sympathetic, the fight or flight branches, or the parasympathetic, rest and digest. Remember, these are the nervous systems that work below the level of consciousness. So these are the nerves that go to internal organs, and they go to glands, like sweat glands and your heart. Motor, skeletal muscle, autonomic nervous system, parasympathetic, rest and digest, sympathetic, fight or flight. Sympathetic nervous system, this is Po from Kung Fu Panda. Po fighting foes is sympathetic. Po eating foe, or pho, whatever it's called, that soup, is rest and digest. So the peripheral nervous system contains two efferent components. Perfect. Skeletal muscle is voluntary. It's body movements. There is an involuntary component to motor, to this branch, somatic nervous system, but that's going to come in with reflexes, like when your doctor taps your knee with that hammer and your leg goes forward. That is somatic nerves. Autonomic nervous system is going to control smooth muscles, cardiac muscles, internal organs. It's involuntary. And there's two main divisions, rest and digest, which is called parasympathetic, and sympathetic, which is fight or flight. No need to memorize all of this, but let's look at a few highlights. The parasympathetic division of your autonomic nervous system tends to slow your heart rate. It slows your heart rate, meaning the more the more stimulus there is from the parasympathetic division, the slower your heart. The less stimulation there is from your parasympathetic division, the faster your heart goes. So there's an inverse relationship. With your sympathetic division, the sympathetic division accelerates heart rate. So you increase sympathetic stimulation, you increase your heart rate. There's a direct relationship between the two. Both autonomic branches innervate the same organ often. So most of the time, parasympathetic and sympathetic, they work opposite directions of each other, where sympathetic promotes you getting ready to fight or run from something scary. So you want to have a faster heart rate if you need to fight something or run away from something. Sympathetic division accelerates your heart. Parasympathetic division generally prepares you to rest and digest. When you rest and digest, you do not need a fast heart rate. So parasympathetic division slows down your heart rate. They have opposite directions. You increase parasympathetic activity, you slow down your heart. You increase sympathetic division, you increase heart rate. So here are the relationships. With parasympathetic, it's an inverse effect. With sympathetic, it's a direct effect. Here's an example of antagonistic control. Antagonistic control, perfect. So one thing real quick. All these nerves, the intensity of the signals that they send are always the same. The intensity, the amplitude of every action potential is the exact same every time. What varies is temporal summation. How often, what's the rate, the frequency at which parasympathetic fibers are sending signals, and what is the frequency at which sympathetic fibers are sending signals. All right, so at rest, your parasympathetic nervous system tends to have the largest effect. Your parasympathetic nervous system tends to have the largest effect on your heart rate when at rest. During light activity, 100 to 120 beats per minute, both branches are working moderately hard. You tend to have they tend to contribute pretty evenly between this heart rate, about 100 to 120. And with high intense activity, the parasympathetic decreases quite a bit and the sympathetic increases a lot. So at rest, parasympathetic dominates. At high activity, sympathetic dominates. Let's compare this to sweat glands. Sweat glands are controlled exclusively by the sympathetic nervous system. Your sweat glands are only innervated by your your sweat glands are only innervated by your sympathetic nervous system. There is no dual innervation. There's no parasympathetic fibers that go to your sweat glands. If you increase sympathetic activity, you increase stimulation to your sweat glands, you increase sweat. If you decrease sympathetic activity, you decrease stimulation to your sweat glands, you decrease sweat. So increase sweat, and like I said earlier, sweat is like salty water. It allows electricity to flow better. You increase sweat, you increase EDA or GSR. So let's see what happens. So we're resting with our eyes open. Heart rate 75, that's pretty normal. EDA 2.57. What do you think is going to happen when you close your eyes and relax? Well, you close your eyes and relax. Your parasympathetic nervous system increases the amount of stimulation it produces, and your sympathetic nervous system decreases. So you close your eyes and relax. You have a decrease in sympathetic nervous system action. You have an increase in parasympathetic nervous system action. Heart rate goes down. EDA goes down. Less sympathetic nervous system action. EDA goes down. Heart rate goes down. You have you have more parasympathetic stimulation. And then your classmates, your group mates scare you. They drop a book on the table. They scream and stomp their feet. They scare you. You decrease parasympathetic and increase sympathetic, your heart rate goes up. You increase sympathetic, your EDA goes up. All right. If you have any questions, please email me and have a great day.